Uh, Welcome to the new session of my interaction with you. So, in this session, I will try to share with you how technologies for genetic engineering or agriculture biotechnology are developed. So far, what you have seen in my previous two presentations that we used available technology in terms of genes which were characterized and validated in groups outside. So the problem of using these technologies is that they are owned by others or their inventors. For example, the P5CS gene was invented by a group in Ohio State University and it is patented by Ohio State University. So it is the property of the organization. Similarly, the tri-1AC gene was codon modified and artificially synthesized by a group in Canada. So when a company approached me to get these products for commercial use, then the question of ownership came, and they were asked to purchase these technologies uh, before they go for commercialization. So there is a bottleneck into this. That is, until unless you own the technology, you cannot commercialize it. And owning a technology can be two ways. Either you invent yourself or you buy it from the owner of the technology. Like intellectual property right comes into play, and it is very expensive to buy any technology from the inventor. So therefore, a lot of efforts people started making in India to develop indigenous technologies so that we don't have to really pay for heavy amounts for commercial purposes. So so it is with this objective, I also started working towards developing technologies, particularly for abiotic stress tolerance. So uh, let me share the slide. So basically, in technology development, we have to go through functional genomics, that is, identifying genes or set of genes who are responsible in controlling a particular phenotype. In this case, say abiotic stresses. So when we look at the cellular responses in response to any stress, you know, the cell responds at the molecular level in four ways. Either it starts inducing new genes in response to a particular stress signal, or it suppresses already expressing genes, or it overexpresses certain genes, or underexpresses a certain set of genes. So this is the four types of molecular responses which are expected at the cell level in response to either biotic stresses or abiotic stresses, for example. So it also constitutes for differential gene expression under different set of conditions. You know. Now, so the target is to to pinpoint the gene or the genes which are getting differentially expressed under a set of growth conditions, and then trying to characterize using transgenic biology for their functions. In other words, we try to link a gene with a phenotype. That is, if you silent that gene, the phenotype is lost of the plant, or if you overexpress, it starts strengthening the trait or the phenotype. Okay, so this is the basic principle uh, behind trapping a set of gene or genes, which may be responsible for a particular phenotype. Now, how it is done, as I explained to you in the previous first lecture itself, that there are two types of approaches to discover new genes or technologies, either hybridization-based techniques or the PCR-based techniques. Hybridization-based techniques basically include cDNA library screening, that is differential screening, subtractive hybridization, microarray-based techniques, and microarrays like DNA chips, either it can be oligo-based or the ESD-based chips, or PCR-based, RAPD, DDRT-PCR, AR-RT-PCR. There are different approaches. So we have in my lab used almost all these approaches to pinpoint differentially expressing genes under different set of conditions. I studied two aspects of this. One is identifying genes which may be responsible for abiotic stress tolerance or management of abiotic stresses like drought, salinity, flooding, And also another aspect which we worked in my group is to understand molecular basis of development. Now, this is another aspect which has not been understood. We still don't know what is the molecular basis of cell differentiation. What are the genes which are responsible uh, for differentiation into shoot or root, So that these kind of questions are still unanswered. So in my subsequent lectures, I will focus on that. But for now, I will focus or concentrate on uh, identifying differentially expressing genes for abiotic stress or salinity stress. And the approach we used is the first one, like uh, making cDNA libraries and then differentially screening this set of library for the presence of unique genes which express under salt conditions or high salt conditions. So uh, what happens in this, we make library into phages. We isolate RNA. In this case, for example, we prepared RNA from maturing panicles or at the latest stage of seed development. Why this was chosen is because during this period, the water is withdrawn and still the plant tries to function normally for seed set and seed maturation. So it was expected that there may be a group of genes which help plant to overcome that phase of seed set and maturity. So this library was prepared and using standard protocols, and then uh, then the library was spread, onto a plate, for plaque formation, So these plaques are then lifted onto in duplicate onto the two nitrocellulose membranes, and then they were differentially hybridized. It's the same set of phage plaques which were taken on two membranes. One is hybridized with the controlled cDNA, that is the normal, and then another set was, another membrane was hybridized with cDNA from stressed plants. So you have normal RNA from the normal plant growing under normal condition, and RNA from plants growing under salt stress conditions. Okay, then these membranes were uh, washed, autoradiographed, and what you see ideally is a scenario like this. So you have various genes which were expressing under controlled conditions will hybridize to the genes which are present here, you know. So, and then you have hybridized with the RNA prepared from stressed plants. So you will see, if you overlap these two autoradiograms, you will see certain common spots hybridizing like this and this, you know, they are common. And like that, there will be hundreds of such common plaques which will be hybridizing to uh, in both the conditions. But then you will see like this, some of them are only hybridizing here, and these are the unique representative of genes which are expressing only under stress conditions and not under controlled conditions. And that's how we end up, you know, identifying genes which are expressed under stress condition and not in controlled conditions. So it means these are the genes which may be responsible for stress management, and therefore they become representatives for further analysis. Okay, so that's how the differentially expressing, this is what is referred as differentially expressing genes, you know, like this and this, for example, you know. Now, once these genes are picked up from here, uh, there, there are approaches, as I mentioned earlier in my very first lecture, through this slide, that these needs to be sequenced to check whether the trapped gene is full length. If not, then uh, the the mRNA is completed. There are different approaches for that. I will talk about that. How, if you have an incomplete mRNA trapped into library, that sequence can be completed. So, but what is important is you need to sequence the gene, you know, and then by doing bioinformatic analysis, you can derive a whole lot of information from that sequence about the putative nature of the gene, what it is and what it may be doing, or whether it is already known in other systems, has somebody already worked. All those things can be determined by doing bioinformatic analysis. And then once you have the gene in hand, what you have to do is to make sure that the gene is expressing under test conditions, or it is really differentially expressing by doing another analysis or real-time PCR. Nowadays, it is the real-time PCR which is used, but when I did this work, at that time, real-time PCR was not available. So only way to check the differential expression was another analysis. And then once you establish that the gene in hand, which one has isolated, uh, it needs to be validated. And validation is done through transgenic approach. So whether you silence the gene or overexpress the gene. Okay. So both the approaches are interchangeably used. Okay. And then uh, one has to find out where this gene product, like the protein, is localized within the cell, whether it is nuclear protein or cell organelle protein or cytosolic protein, or it is membrane protein, where it is localized, and very importantly, what is the function of this protein. So all these things need to be, in a systematic way, needs to be studied. Okay. So with this kind of introduction, and as I mentioned, we approached the technology development for abiotic stress tolerance by differentially screening cDNA library prepared from panicle, maturing panicles of rice plants. We ended up in picking up a series of genes And I will discuss one such gene which we finally decided and took it to its logical end. Okay, so when we picked up one such gene and sequenced it, got it sequenced, you know, and we did a bioinformatic analysis, you know, of the sequence, you know. So what we found out, and by this time, the rice genome sequence was already in place. And a lot of ESD databases were generated for rice. As I mentioned, rice is one of the crop which has been worked upon very heavily ever since the improvement work started, like either through mutation breeding or through newer approaches like the genetic engineering, for the simple reason that it is the staple food for millions of people globally. Okay, so people want to improve its productivity both qualitatively and quantitatively. So a lot of background data is available, and today, a whole lot of bioinformatic data is available. So when you take a sequence and blast it, you know, you can generate information. For example, in this case, you know, you can see the, you have the uh, translation start site and then the poly A tail. So from end to end, the sequence seems to be complete. And when we look for the coding region, you know, it has 516 base pair coding region starting from ATG to this. From here to here is the coding region. And from here to here is the non-coding region, basically. Okay, and this 516 base pair translated into a protein that is again by doing in vitro translation, which codes for in this pink region, you know, which is the coding region, translates into a 171 amino acid protein, you know, and further, when we try to annotate this sequence onto the rice chromosome, it is it is located on chromosome 6 of rice genome, and it has two introns. You know, when we look for intron exon composition, and these introns actually is beyond this part, so it is 5' upstream region from the coding region start site. You know, so those were missing, but they were redundant in the sense that these two introns in the untranslated region. So what is important is the translated region or the coding region, you know, which is involved in making the protein. You know, to that extent, we could drive the information about the gene sequence. You know, another thing which we could find out that it is a putative zinc finger protein, you know, zinc finger protein, uh, and further information on the zinc finger protein, basically, uh, it is in rice. It's a gene family which has 18 members, okay? And it is referred as Oriza sativa stress associated protein gene family. In short, OSSAP1 to 18. So you have Oriza sativa stress associated protein 1 to 18. So there are 18 members of this gene family, you know. And these gene family is characterized by the presence of two functional domains, what we call zinc finger AN1 and zinc finger A20. ZNF AN1 and A20 functional domains. So this is the characteristic of this SAP gene family, you know. Now, in another group in India, uh, all the 18 members were looked at for their differential expression by using real-time PCR. And what they found that all these members were multiple stress inducible genes, you know. So, and the same group actually worked on the first member of this gene family, which is SAP1, OSI, SAP1, Oryza sativa stress associated protein 1, you know, and they demonstrated in tobacco that if you overexpress this gene in tobacco, it confers tolerance against salinity, drought, and cold uh, conditions. And this paper appeared in PNAS in 2004, actually. And when we looked at the gene which we isolated, whether it is SEP1 or what is that member, so we discovered that this is the SEP8, that is eighth member of that gene family. So we named it as uh, uh, OSI SAP8, you know. So SEP1 was demonstrated in tobacco, and then we started looking at the SEP8, you know. So just by doing bioinformatic analysis, we could find out that it is a gene, unique gene, which has not been worked upon, and also may have uh, a role in stress management because it is differentially expressed in the under stress conditions. You know. So further, when we look for the domains in this sequence, translated protein sequence, we found out that the SEP8 gene also has the characteristic two domains which I referred to, ZNF A20 and AN1. And this is the 172 amino acid protein in silico translated protein sequence. So it had these two domains. So what are these two domains? Actually, they are the domains which are found in the proteins from animals. For example, ZF A20. Now, this is seen in the animal cells, or it is responsible for inhibitor of cell death, like okay, and it has four cytosine actually. This domain has four characteristic cytosine and interspersed with any other amino acid, you know, but it has these at these positions four cytosine. And then ZFAN1, this is uh, located at the C-terminal end of the protein. This is on N-terminal end of the protein, and this is the C-terminal end of the protein. So A20 is on uh, N-terminal of the protein, and this is on the C-terminal protein, you know. And it has C6H2, you know, characteristic amino acid constitution of this domain, and it is present in a protein in the xenopus levels, you know. So these the proteins which have either one of them or both of them are present in animal systems. None of the protein which has both these domains were reported from plants. So this was the first protein which has both these domains in the uh, in the same protein. So after knowing or after deriving this kind of information, we went on to do some wet lab work. And first was to check whether whatever we have fished out from library is part of the genome. And that is done by certain analysis, you know. So this is the coding region of the uh, SAP8 gene from here to here, and it has two unique sites. One is Hindi 3 and another is UPAR 5. So the genomic DNA prepared from the plants was digested by these two enzymes. So if you cut with Hindi 3, it is going to generate one fragment this side and another fragment this side. Similarly, EcoR5, it is going to generate, means it will cut one in the gene and another cut will be in the genome this side and another will be in the genome in the other side. So it is going to split this gene into either this half, these two halves, or this one and this one and this one and this one. So and then if you have a gene which does not cut those anywhere, like BMH1, it doesn't cut. So it is going to take the whole thing in their fragment. BMH1 will genomically cut on either side of the gene. So, and then you run it on a gel and take it on membrane and hybridize with the whole probe from here to here. It is radio level probe from here to here. So when this, it is going to hybridize to this fragment as well as this fragment generated by Hindi 3. Similarly, EcoR5, this fragment and this fragment. So it is going to generate two hybrid spots, like here you can see 1 and 2, 1 and 2, and BMH1 generates only one, you know. Uh, so it means whatever sequence I was referring to, or the gene we picked up from the from the library is part of the genome. That is very, very important to establish that. Otherwise, it can be experimental artifact as well. Okay, after this, the point to be looked at is whether this gene is differentially expressing in plants. That is very, very important aspect for genes which are differentially expressing. So if you see here, this study was done in seedlings of 8-day old. So you have seedling grown under normal conditions, and then under different stress conditions, for example, sodium chloride, mannitol is used to mimic the drought conditions, abscisic acid and salicylic acid. Okay. Why abscisic acid? As you know, it is the stress hormone of plants, and salicylic acid is the one which is responsible to give biotic stress resistance to plants. So these four factors were checked. What happens. So if you see here, this is the control plant grown under normal conditions. And when the seedlings are grown under high salinity, you can see a lot of transcripts getting accumulated in presence of sodium chloride, mannitol, abscisic acid, and also salicylic acid. So it means this gene is overexpressing itself when plant is exposed to different stress conditions. Okay, so that's one part. So it means that the gene is differentially expressing. And also when we look for whether it is in the roots or in the shoots. So you can see here also that shoots expresses more of it as compared to root. Okay, so it is showing overexpression in a organ specific manner. So, and this is the actually study done 7-day or 8-day old seedlings, and then they were exposed for 24 hours to these conditions, and RNA was extracted, and then hybridization was conducted. Okay, now, but if you have a scenario like this, you just cannot conclude that it is getting induced. It may be the transcripts may be getting accumulated for more than one mechanisms, or by following more than one mechanism. So it is very important to establish that the synthesis is getting increased in presence of these signals. and for that, a time kinetics need to be done. And that's how it is done here. If you see under salt conditions, if you see 0 means before the start of the stress conditions, then 2 hour, 4 hours, 6, 12, and 24. So that's how, the RNAs were extracted at different times, run on gel, transplanted, and then they probed with the cDNA of SAP8. And this was done in the different stress conditions like drought, desiccation, heat, submergence, wounding, cold. So different abiotic stress conditions were tested, and they were checked that what happens to the synthesis of the transcripts of this gene. And as you can see here, this is the control 0 hour, and then 2, 4, 6, 8. Up to 12 hours, there is a continuous increase, and then slightly the amount goes down by 24 hours. So the same thing is observed for drought. There is a continuous increase under all the conditions. Desiccation, if you see, and heat stress, you see, it is more at 24 hours. And similarly, you can see submergence and wounding. Wounding, you can see within 2 hours, you see more transcripts here, which goes down with time, So similarly, cold treatments, you see here, heavy metals also, this gene respond to heavy metal there too. If you see control 3 and 6 hours, and with this, if you compare zinc, copper, magnesium is lethal within 3 hours. The plant dies, And then the cadmium, you can see a lot of transcripts are accumulated in 3 and 6 hours as compared to control. And similarly, you can see these other metals, which were tested. But then the question was, what happens between 0 and 2 hours? What is the quickest time on which the gene is overexpressing? So we try to go down, say, between 30 and 60 hours. So this was done through RT-PCR. And you can see here that within 30 minutes of the signal given to the plant, um, 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 given to the plant, it starts synthesizing under all the conditions. Okay, so the conclusion of this part is that the gene is maybe an early induced gene during various stress responses, and it may have a role in the very early signal transduction activities, or in other words, the northern analysis, what it indicated that this gene product is involved in early, very early activities of signal perception or signal transduction. Okay, so so now it means we have a gene in hand which is differentially expressing under different signals. And so we have a candidate in hand, basically, which can be further validated for linking or for its linkage with a phenotype, in this case, say abiotic stress tolerance. So I will stop this discussion here and will continue the next part of the work, that is how it was functionally validated in the using transgenic biology approach. So thank you very much, and we meet again in the next session.